So my name is Dirk Arnold. I'm medical director of the Asklepios Tumorzentrum in Hamburg in Germany, which is a kind of network cancer center. I'm involved now for many years in colorectal cancer research, mostly clinical research, having been the chairman of the German AIO group and as well as the EORTC GI cancer group for some time. This gave me some insights in clinical trials, and I'm still following the field with great interest. The treatment landscape is evolving in, let's say, two ways. First is new drugs are entering the field and allowing specific treatment for whatever molecular subgroups. Some examples are specific RAS inhibitors or now BRAF V600E mutation inhibitors, etc. This is unfortunately so far only relatively small subgroups. But the other improvement is clearly to improve the therapeutic principles, which we already address in colorectal cancer, which is anti-angiogenesis and also anti-EGFR inhibition. And we see more and more drugs entering this field also and trying to improve the standard of care, chemotherapy plus one of the two antibody groups. OriGAMI-1 is, let's say, the signal-seeking trial looking to 3 to 5, somehow differs, cohorts of patients with whatever pre-treatment or molecular subgroups, aiming to identify whether Amivantamab does work in this group, whereas OriGAMI-2 and OriGAMI-3 are large randomized trials trying to set Amivantamab as the standard of care treatment in comparison to standard of care first-line treatment, that's OriGAMI-2, or second-line treatment, that's OriGAMI-3. We had some let's say, decent looks on now subgroup analyses from OriGAMI-1, we'll say, from the clinical trial with the 5 subgroups, and specifically the focus has been on biomarker analysis, looking on tumor gene expression and immune cell changes in this group of patients with refractory metastatic colorectal cancer, which underwent treatment with Amivantamab, and giving, let's say, the mechanism of action of Amivantamab with these 3 different mechanisms of action. The expectation has been high that it could be interesting to identify a subgroup of patients which does benefit in a specific way from the treatment with Amivantamab. It has been a really scientific arena where OriGAMI-1 was running. And the key findings of this biomarker analysis, next to the great efficacy which we have seen in all of the subgroups of EGFR pretreated, non-pretreated, left-sided tumor primary, right-sided tumor primary, etc. These have been the clinically subgroup with more or less similar efficacy and similar promising efficacy in all of the subgroups. But specific, when we look to this biomarker group and this biomarker analysis, we have seen that Amivantamab increases the cytotoxic immune cell signatures, which is somehow consistent with immune cell infiltration into tumors. And this downregulates the EGFR and cell cycle pathways. So therefore, we have seen that the mechanism of action by blocking the anti-EGFR pathway and the anti-MET pathway is also preclinically confirmed. And this gives us some more confidence in that the observations are not only by chance observations, but are really with a solid biological background. And this gives real confidence that the randomized phase 3 trials, OriGAMI-2 and OriGAMI-3, have a good chance to be successful. The interest in OriGAMI-2 and 3 is really high. These are globally recruiting randomized trials. And I wish to encourage further recruitment to these trials. So we're really recruiting with a high speed. Both trials, each of them in first and second line, belong to the largest trials ever in colorectal cancer. And it would be great to have this, let's say, done and recruited by mid of next year. And it looks really promising that we will be successful with this. The first line trial is more than 1,000, so slightly more than 1,000. And the second-line trial, I don't have the exact numbers, is between 600 and 700. So also a relevant number of patients there. So these will be really big phase 3 trials. And next to the clinical readouts, this big number of patients is super important because this allows us to learn from the molecular subgroup analysis and to all of the, let's say, translational research testing to this big cohort of patients. So therefore, it's important that we put some speed behind and that we have a large number of patients, large number of tumor specimens, ctDNA, etc., that we can really do the best translational research to this.